So first, I'd like to thank the organizers for the opportunity to speak today. I'm a graduate student in Katie Pollard's lab, and we're in the Gladstone Institute of Cardiovascular Disease, and my thesis project is based on identifying functional regulatory variants that might be associated with developmental disorders with cardiovascular phenotypes. So an important part of that question is going to be to identify a strong set of developmental heart enhancers. And that question can actually be broken up into two sub-questions. So first, in predicting developmental heart enhancers, an easier first step is where are generally developmental heart enhancers? And then second, can we ascribe tissue specificity? Are they active in the heart or in other tissues? And the second question is, which data actually helps find these developmental enhancers? So with thanks to the ENCODE project and other large data set studies, we've found many clues that can help identify enhancers. We see here enhancers are marked by proteins bound such as P300. We know that there are certain histone modifications associated with enhancers. Also important to the question of what is an enhancer, though, is what is not an enhancer. We know that certain proteins like CTCF are going to be bound to regions that probably aren't enhancers. Additionally, we know of several sequence motifs that underlie known developmental enhancers. So an overview and some punchlines of this talk. So I'm going to present a novel machine learning approach that we've developed that both predicts tissue-specific developmental enhancers and also evaluates the utility of over 500 different data sets, such as those in the ENCODE data set. So when I say tissue-specific developmental enhancers, heart enhancers are the ones I'm most interested in. However, we can also do this with any set of tissues that we see in our training data, which I'll show you in just a moment. So here's a quick spoiler alert with the results of this project. We were able to predict a lot of novel enhancers. We validated them through a transgenic mouse assay, and we'll also show you how nearby genes are enriched for relevant functions. So we have some interesting genomic context in these predicted enhancers. And the answer to what is the most informative data out of all of this sea of data in the ENCODE project and such, to answer the question, is this region an enhancer, we see that accessible chromatin in many different data types can inform that question. And to answer in which tissue is this enhancer active, it's actually more important what the cell type that data is coming from than the mark itself. So the machinery of our approach is based on the idea that you can characterize every genomic region with regard to chromatin, protein binding, and sequence motifs, thanks to all these genome-wide data sets. And we have it from many, many different cell types. And as you can imagine, for a question like mine, finding developmental heart enhancers, a lot of the ENCODE data isn't going to be exactly right. However, we have some good examples of what do these enhancers really look like. So these top two examples are from the Vista enhancer browser, and that's what we're using as training data in our algorithm. And Len introduced the Vista browser yesterday in great detail, but I'll just remind you, thousands of putative enhancer sequences were tested in this transgenic mouse assay, and some were shown to have enhancer function at embryonic day 11.5. Others were not. We're also, in this approach, we're characterizing regions of the genomic background. So we know what is the chromatin state, the protein binding, and the sequence motifs for true enhancers, regions that were tested that aren't active at embryonic day 11.5, and also regions of genomic background. And we can put all of this into our machine learning algorithm that then determines the underlying feature composition to, one, define enhancers, and two, ascribe that tissue specificity. So here's the basic pipeline that we use to predict if a region is a tissue-specific enhancer or not. So we start off with regions of the genome, and we can ask, all right, is this region a developmental enhancer? And then if the answer is yes, we can then go on and ask, is it a brain enhancer, a heart enhancer, or a limb enhancer? So I'm going to spend a little bit of time right now discussing the performance of the algorithm on this first step when we ask the question, is this region a developmental enhancer? So the main punchline is that more data equals better predictions. We started off by using just the most relevant or seemingly most relevant ENCODE data, and that is to say, since we're looking for embryonic day 11.5 enhancers, we looked at the embryonic data in the ENCODE project. There's about 70 data sets that we included here, and we were able to get a performance of 0.73 AUC, and that's how we're quantifying our performance. And again, we've seen a lot of ROC curves, but just a refresher, if you're skating along this line right here, you get an AUC of 0.5, and that's no good. And if you're all the way up here, you get an AUC of 1, and that's spectacular. So our first try with the embryonic data, 0.73, okay. We then decided to add the embryonic stem cell data. So we added about 170 more data sets. And these are less actually relevant to that developmental time point we're interested in. But there's 170 more data sets, so now we're up around 250 data sets. And this includes a lot of information about what might be poised, what might be about to turn on in that developmental time point we're interested in. And we see that our performance improves quite a bit to 0.83. We then decided to add a lot more data. So over 500 data sets here, including adult cell lines, for example, cardiomyocyte cell lines, lots and lots of data that, again, not directly relevant, but might contribute a little bit to our ability to call developmental enhancers. And again, we see that that does improve our performance. But you see that it's a smaller improvement. We go from 0.83 to 0.87, and we practically double the data set. So the less relevant the data set, the less it informs it, but it still informs that prediction a little bit. And last, we added the sequence data. So here, those the Kamer SVM that Mike introduced yesterday. It's very similar to the way that we looked at our sequence here. So we incorporated all that, and we were able to get a strong classifier that performed at 0.91. So shifting to that second question now, are we able to ascribe tissue specificity? We see that some tissues are easier to predict than others, and we see here that heart enhancers are easier to predict than others. There's a lot of ideas I have about why this might be, but the main driving feature appears to be the fact that heart enhancers, as Len mentioned yesterday, are less conserved than other enhancers. We see that less conserved enhancers are easier to predict than more conserved enhancers. And again, heart at embryonic day 11.5 is relatively more developed than brain and limb. A lot of the mammal-specific features we see, like the ventricular septation into that four-chambered heart, is happening right at this time, at embryonic day 11.5. So we ran this across the whole genome, and we predicted about 84,000 developmental enhancers, and we were able to ascribe tissue specificity to about 30,000 of them. And what's interesting, you see here, is that the developmental enhancers that we predict are active mostly in a single tissue. About 70% of them are unique. And that's not really surprising what we know about enhancers. They're tissue-specific generally. But we do see some overlap. So they're not all entirely unique. We validated several of these in a transgenic mouse assay. And I'm showing you one example here of where we predicted it to be a developmental enhancer. We predicted the heart and the limb activity, and we see that we do have that heart and limb activity. We didn't test it for neural tube, or we didn't run the algorithm to look for neural tube activity, but we do indeed see a fairly complex expression pattern here. As a negative control, we also tested a region that we predicted not to be a developmental enhancer. We had a strong score that this was not a developmental enhancer, and we did indeed see that there was no expression at embryonic day 11.5. Putting this in a genomic context, we did several computational analyses to test what genomic context are we in. So we see that the genes near heart enhancers are enriched for expression in both cardiomyocyte and heart. And we see the same similar pattern when we're looking at brain. That's going to be enriched near the brain tissues, and limb near the limb tissues. And this, again, was with the GNF Atlas data. Next, we used the GREAT tool out of the Bejarano lab, and we're able to see that the genes near our heart enhancers are enriched for heart functions, which is great. So we see artery morphogenesis, artery development, TGF-beta receptor signaling pathways. And again, we see similar patterns for the limb and for the brain enhancers being enriched near functions that are relevant to those tissues. And lastly, following on the idea that GWAS SNPs are found in functional regions, and that they might be marking regions of functional variation in the non-coding parts of the genome, we overlap them with our predicted enhancers, and we find that we do indeed see a significant overlap between our enhancers and the GWAS SNPs. We see this significant overlap in both the heart and the non-limb, non-brain, non-heart regions. We see a similar trend with the limb and the brain, but it's not significant. So now to address that second question, which of all of these 500 features in ENCODE that aren't exactly right for our prediction question are really informing this enhancer predictor? And we see that the top weighted features are generally marks of open chromatin. So DNase hypersensitivity, FAIR, digital genomic footprinting. This is a summary of the top 25 or so weighted features, and they're coming from many different cell types, but similar marks keep popping up. And not surprisingly, we also see H3K4 monomethyl and P300 coming as very informative marks of where we might find a developmental enhancer. And to ask the question of what data is informing these predictions of tissue specificity, we see that it's actually more important that the cell type that the data is coming from is relevant to our question. Here again, we're looking to ascribe heart specificity to our predicted enhancer, and we see that in partially differentiated cardiomyocytes, we get a lot of powerful data off of both open chromatin and then this other mark, H3K4 trimethyl, which we don't necessarily associate with enhancers, but that's again supporting the idea that the cell type is more important than the mark. And we see that embryonic cell data is also very informative, and again, that's probably marking poised or about to be ready enhancers. So just to wrap up, I've shown you how we have many novel enhancer predictions. We're currently doing further in vivo validation in both mouse and zebrafish, so we can start to get some quantitative performance scores for how well we're actually doing in vivo. And I showed you how we've analyzed the utility of each data set, and so far we've just looked at the human data sets, and next we're going to look at mouse and other species where we have a lot of data that might inform where especially deeply conserved enhancers might be. So we'll be evaluating whether mouse data can be used to help us predict human enhancers. And then lastly, as I mentioned when I showed you the figure with the GWAS SNPs, we're really interested in the functional variation that we find within enhancers, and we're currently investigating how these might relate to several different cardiac phenotypes. And so I'd like to thank the whole Pollard lab, specifically Katie Pollard, she's a fabulous mentor, Tony Capra, he's a postdoc I've worked very closely with on this work, and Rebecca Trudy for some awesome discussions. Also, big thanks to True Wynn and Bruce Conklin's lab, who taught me how to clone and do the molecular biology. You might have noticed I'm a computational biologist, and that probably wasn't an easy job for her. And now I'd just like to thank you for your attention.
Question. Thank you for a very nice talk. I have two questions. So the first one is, I'm quite surprised, or I mean, you know, I'm curious as to how you continue seeing increasing power as you keep adding all these potentially irrelevant data sets. So I'm curious about the way that you're evaluating your performance. In other words, are you training your classification on one training data set and then leaving out a holdout data set?
Right, so we're doing tenfold cross-validation, where each time on this one, each time we're holding out 10%. In other iterations where we held out 50%, we see similar performance.
That's interesting.
Yeah,
so
it was definitely surprising to us too. Even when we did throw every single ENCODE data set in that, we did see again a marginal improvement. But again, keep in mind we're adding hundreds more data sets and getting only slight improvement.
Yes.
So
I mean, it is kind of interesting. I mean, one possibility might be to cluster beforehand to make sure that you're eliminating these redundant data sets that might be giving you power even when you do these tenfold cross-validation.
So the way that the SVM is, we're using a multi-kernel SVM, and if a data set is actually redundant, then that piece of information is already contributed, and so if another data set is contributing that exact piece of information, it won't get a high weight, and it won't really improve the performance. So with that regard, actually our data sets are not independent.
Not on the level of data sets, but on the level of examples.
Oh, you mean if we're training just on a subset of the Vista browser?
Yeah, so basically if you cluster your examples first to make sure that they're as non-redundant as possible.
Like cluster them on tissue type?
Sure. We can talk offline. The second question was about the DNase hypersensitivity. Basically, the marks that you were showing, P300, H3K4ME1, were all enhancer marks, and then DNase hypersensitivity is found both at promoters and enhancers.
Right.
I'm curious as to whether, as a class, you considered DNase specifically away from transcription start sites.
So we have done that where we've looked at specifically where we've excluded regions within 5 kb of a promoter or of a known transcription start site. We get similar performance, but we predict fewer enhancers. But again, we're just trying to mostly recapitulate what's in the Vista enhancer browser of the known enhancers, and since several of those do fall within
promoters.
Right.
Okay, so I mean, it is possible that in your training set you actually have promoters, and again, I think this came out during Len Panakia's talk about sort of whether it's an artificial separation that we make between enhancers and promoters.
Right.
Thank you very much for your talk.
Thank you.
for this support vector classifier, you need examples of true enhancers and true non-enhancer regions. How do you define those?
So that's a good question. So I mentioned that it's a two-step classifier, and I didn't really go into much detail. But in the first question that we asked, is it an enhancer or not, we used any region that was tested in the Vista enhancer browser that showed any activity, any enhancer activity as the positive. And then we used random genomic background as the negative. And it's not a true negative data set because it might contain some enhancers that no one's ever found. But it's a good proxy. And then for the second question, we then asked in a very tissue-specific manner. So in this case, we'd be able to say, this is a limb enhancer, this is certainly not a limb enhancer. And so we would train on all the Vista enhancers that had limb activity for the positive, and then our negative would be the regions that were tested in Vista that showed no limb activity at embryonic day 11.5. Thank you.